Hello and welcome to this part of the FAST tutorial series. In today's video we'll be looking at how to use overlays to provide information-rich visualizations of our datasets. So if you watched the previous video, division detection, you probably saw a sneak preview of the overlays module, but just to start at the beginning, to open up the overlays module you just click this button here, which should turn green as soon as you've finished either the tracking or the divisions module. And when you click that button, you should get this screen pop up. So here we have the first frame of our movie, which is the same as we were looking at in the previous video. So that's this video of some cells dividing over time. You have these options on the left-hand side here. Now these options define the setup of the overlay. So we have, for example, the underlay source drop-down menu. So this allows us to choose which of the images we want to use as our underlay. So we have channel 1, which in this case is the phase contrast image, but we also have our segmentation, which we can choose. And if we had fluorescence data, that would also be present within this drop-down menu as separate channels. So we'll stick to channel 1 for the time being. We can also choose the overlay type. At the moment we have no overlay, but we can choose, for example, tracks. We can move to a later time point using this slider and edit box here, just to see what these tracks look like. And so you can see that we have these tracks appear which indicate the movement of the object's centroid over time from early time points to the current time point here. We can also have ellipses, which provides us with a elliptical reconstruction of the object geometry, and also provides us with an arrow indicating the motion of the object between this time point and the next one. We can have masks, which just provides the segmentation as a mask overlaid on top of our image. And then we can also choose boundaries, which is very similar to masks, except that it hollows out the interior so that we can see what's going on inside the cell. So those are the 4 different overlay types. We can then choose which of the different types of information we want to overlay. So at the moment we just have raw, which provides this kind of constant orange value over every object in the image. But we can choose, for example, track IDs. So this colors every object according to its index. And so you can see as we move through different time points, these colors remain constant, but then they change as cells divide. We can also choose this lineage option, which colors cells according to their lineage. Obviously, this is only available when the division detection module has been run. But what this does is it colors our 2 maternal cells and then retains those colors over time so that we can see all the descendants of those 2 maternal cells at the later time points. And finally, we can choose this data option, and this allows us to visualize any of the data that we have either collected using FAST or applied ourselves into the track data structure. Now there's some tutorials on the wiki about how you can apply your own data to that track data structure. For the time being, I'm just going to focus on these automatically generated fields, and those are available in this drop-down menu here, so this data selection drop-down menu. So for example here, the default is X, so objects are just colored according to their X position. We can also color cells according to their Y position. That's not terribly helpful, but it can be kind of attractive. You get this nice flag effect. More useful are these other fields of the track data structure. So for example, we can see how large different cells are in different parts of the colony. We can see what angle they're pointing in, so whether or not there's any kind of orientational structure to our data set. We can also see their age relative to their birth time, And we can also see what I call their generational age. And again, this is only something that's available because we've run the division detection module. So these colors here represent the age of cells in generations since those maternal cells. So you can see if I scoot this back to the beginning, you can see that these two maternal cells have a color corresponding to 0. But if I move this forwards, you can see that we have a range of different ages at later time points. And so this allows you to kind of see, for example, these cells have stopped dividing, and so you can see that they're stuck at an earlier generational age. Final drop-down menu is the data color map, and this allows you to customize the coloration of your dataset. So the default here is the jet color map, but you can choose any of MATLAB's default color maps. So we have Perula, for example, that's the kind of classic MATLAB color system, shading from blue up to gold. You can also have pink, if you're into that kind of color, I don't know. So yeah, I mean, you can really customize exactly how you want to visualize your data to maximize its kind of aesthetic appeal. The final option that we'll discuss with this dataset is this checkbox here, which allows us to show the ID of every object in the field of view. This is useful if you want to focus in on a specific cell, either in the plotting module, and we'll see some examples of this in the next tutorial, or if we want to look at the raw data in the track data structure. So for example, if I see that there's something interesting going on in this cell here, I would just click the Show ID button here, and that would tell me that this is cell number 65, or track number 65. And then I could use that information to isolate that data within the track data structure or the plotting module, as we'll see. Okay, so that is how we set up the overlay for a single image. If we now want to apply this to the entire data set, so let's just set this up so that this is Y. Let's take off the indices. So to apply this particular setup to the entire data set, we would just click Run Them All, and then we would see this new folder appearing within our root directory called Overlays, and this contains all of the images with this set of overlay settings applied. And then we can use these to create a video of our dataset with the overlay settings applied. So here you can see this is one that I made earlier. To generate this, you just use the file import image sequence option, as you can see on the wiki. And you can see that we have colored all of these cells according to their Y position over time. So as you can see, you can build up really quite complex visualizations of your dataset using this module. So one last thing about the overlay module that I'd like to discuss just before we finish this tutorial is this box here, which is grayed out, called the mark events box. Now, as you'll see on the wiki in a lot more detail, FAST has this idea of phenomena called events. Now, these are things that occur at discrete points in time, and the nature of these phenomena varies very much depending on the research question that you're interested in. So perhaps you're interested in figuring out when cells reverse their direction of motion, for example, or maybe you're interested in when they grow above a specific threshold. So you can measure these events using automated post-processing techniques. This is a script that I've written that allows me to do this, called label threshold events. You can get this on the wiki in the post-processing toolbox. And it does various tasks, but at the end here you can see that we assign the measured events to this field called proctracks.event. Now, what I've done is I've applied this script to this data set here. This data set is from a study looking at the type 6 secretion system. You can find more details in the original paper, which I've linked in the description. But the basic idea here is that we have these discrete foci appearing within our fluorescence channels, and then doing something and then disappearing. So basically what I'm interested in is knowing when these foci appear. So you can also see that there are two different colors of foci. So we have these green foci And then we also have these red foci down here, which correspond to two different species, both with this system. So I've applied this script to this dataset, and if I now close down the overlay tester and move to this other dataset, then if I now go to the overlays module, you can see that this mark events button is now ungrayed. So I'm going to choose channel 2, so that is one of these two channels with the fluorescent spots available, and let's move to a reasonably late time point. And so you can see here that we have these two different colors of ring when I check this box. So the red rings indicate time points where this has detected firing events in the green channel, so that's this channel that we're currently visualizing. And so you can see very faintly that there's a firing event going on there, and then cyan circles tell you when it's occurring in the other channel. So if I now switch, you can see that we have these faint foci appearing within the other channel as well. And so you can imagine that you could then use this overlay module to test whether or not your event detection is working properly. Allowing you to visualize, in this example, where these foci are, and then matching that with the event detection, but depending on your research question, obviously that will vary. So that ends this tutorial. In the next tutorial, we'll be looking at more detailed plotting using the plotting module. So looking forward to seeing you there.